in our Thank you for your interest in our poster titled A Review of Problems with Pharmacovigilance Programs in Biologics. The reason we decided to do this work was the recognition that post-approval experience in the clinical setting is becoming increasingly important as the drive to speed drug approval process grows. For biologics, this has become an important issue because of the abbreviated process for approving less expensive biosimilars, which relies more on analytics than clinical trials, as shown on this pyramid diagram on our poster. A strong pharmacovigilance program depends on the accuracy and completeness of reports, including the identity of the drug being associated with the event reported. There are several ways to identify a drug, including the brand name, the lot number, non-proprietary names, machine-readable coding, and other identifiers that are often used for billing. Each of these methods has its advantages and disadvantages, but a strong program will rely on multiple identifiers in case one of the methods fails. This is based on a well-recognized principle used to identify and prevent hazards from becoming losses in the industrial engineering field, as shown in the Swiss cheese model in our poster. One identifier that has received attention, but for which variability exists around the world, is the non-proprietary name of the drug. For biosimilars, a different non-proprietary name is necessary because these drugs, while are not identical to the reference product, even though they're similar, and a different clinical outcome may be observed. While a method to distinguish among biologics and biosimilars exists in the US and some other countries, it does not exist in other countries. A global standard would be very helpful in strengthening the pharmacovigilance system worldwide. Our review of the literature is in this poster. And for example, an early 2013 study of biosimilar identifiability in the Vigilance ADR reports found that biologics were identifiable in between 76 and 96% of the time, depending on the product. A 2014 survey of Italy's spontaneous reporting system found identifiable brand names was indicated in 94.8% of the biological related reports and 98.7% among the three categories for which there were early biosimilars. A 2018 review of the Vigilance ADR reports for infliximab revealed that 35% contain no brand name. The 2019 processes ranged from 79% to 91%. But among ADRs reported and analyzed, only 38% had an identifiable brand name. A review of the 2020 Canadian ADR reports for infliximab contained the brand name only 63% of the time. There are now 7 approved biosimilar products for infliximab in Canada that share the same INN because they don't require a distinct INN in Canada. When we surveyed those who submit reports, namely the physician, in a 2019 survey of 579 European physicians, we found that 84% included the brand name in the adverse event report, and 61% provided the manufacturer's name, and 72% identified the biologic only by its non-proprietary name. The Canadian ADR report analysis is consistent with a 2017 survey of Canadian prescribers, 403 prescribers, that revealed that an average of 30% of prescribers do not include the brand name in adverse event reports. 26% use only the non-proprietary name, and 23% consistently use batch numbers. Finally, in a WHO report titled Regulatory Challenges with Biosimilars, an update from 20 countries, it notes the lack of consistency in the nomenclature of biologics and biosimilars causes concern about prescription mix-ups, unintended switching, and traceability. The authors state it is clear that naming and labeling are both very important for the identification of products and also for pharmacovigilance and prescribing. So based on this review of the literature and the surveys that have been reported, we conclude that because biosimilars are not exact copies of the reference molecule, it is important to distinguish among the different biologics, both reference products and multiple biosimilars. Adverse drug events are often incomplete, as we see in the literature that's been published, and missing information about the exact identity of the drug whose reaction is being reported is a challenge. Brand name and batch numbers are helpful in identification, but these are not always used in reports or in the clinical record, as we have seen. Having distinguishable non-proprietary names for reference products and biosimilars would create another layer of safety, as we see in the Swiss cheese diagram, in identifying the medicine that the patient actually receives and is associated with the event that's being reported. Clinical experience gathered from the increasing use of biologics and biosimilars is more important given the declining role of clinical studies that are needed in the abbreviated biosimilar approval process. And finally, this experience will enhance prescriber and patient confidence in biosimilars, speed the adoption of these less expensive medicines, reduce healthcare costs, and increase access to these important treatments. Again, let me thank you for your interest in our poster, and we are pleased that you've chosen to observe it and listen to our video. Thank you.